analysis of epigenome data from quite a different perspective than the previous. See halfway through the talk, the ENCODE data is also. So our medium-term, long-term goal is association studies and marker development. So what's already happening in genome-wide association studies field, experimentally, but even more so bioinformatically able to case control studies, epigenetic case control studies with participants, which is of course a big challenge of the exponential mapping, also in terms of the data analysis. So to put the analysis that I'll be presenting into context, I'll give a quick introduction into the role of epigenetic defects in cancer, but if you're interested in other diseases, epigenetics seems to be related to very many diseases. But in cancer, it's particularly well known that epigenetics provides a way by which tumor cells rid of their tumor suppressor genes in order to enable or unlimited growth, and that in fact until a few years ago the view has had prevalent reasons of genes and cancer is genetic damage, but that epigenetic activation is, which has important implications because when you have tumor suppressor gene entirely in terms of DNA damage, it's very difficult to reactivate this because it's just not there anymore, it's not functional anymore, which is why classical chemotherapy or therapy uses essentially an approach to poison cancer cells at a dose that doesn't poison the human or not more than necessary. Whereas in terms of epigenetic alterations causing cancer, there's a much more promising because epigenetic aberration reversed such that the cancer cells will then normal cells again, or at least they will undergo up. This has already been shown to work. There are a handful of FDA-approved epigenetic treating leukemia diseases such as myelodysplastic syndrome, but by and large we don't really understand how epigenetic drugs work. So the first talk will not be so much on treat cancer by genetic drugs, but on DNA methylation mapping to which is also described as the advantage of epigenetic alteration that they development that they genetic defects, and even from the blood of cancer to detect alteration, either because both the blood to the same environment stimuli, which would be like direct and the tumor, or direct tumor and shedding the biomarkers have been a very active area of research for the last 10 years, in particular for the last 3, 4, 5 years. And now there are a couple of showcase examples that have been established, especially based on DNA methylation. So the SAPT9 promoter methylation assay is being sold commercially already as a way of early detecting colon cancer based on that sample. Another example is the MGMT gene, which predicts chemotherapy in brain tumors and is also widely used, for example, at the Massachusetts General Hospital. It's routinely used when selecting the chemotherapy for glioblastoma patients. Then as a third example, in addition to the diagnostics and therapy selection, the therapy optimization. When you treat patients with drugs that cause demethylation and you want to monitor the effects that the drugs have on the organism, you can use optimization biomarkers to provide feedback and a more personalized approach to medicine. Actually, 2 out of these 3 biomarkers have already been detected by relatively unbiased in terms of finding genes, and that by carrying out systematic epigenetic studies, we can discover a substantial number of additional biomarkers with a role in cancer, but also in other common diseases. The basic step is all that you start with a case control cohort, and you perform DNA methylation mapping or epigenetic of the cases and controls, and then by comparison to derive a list of candidate biomarkers. The challenge that emerges is to efficiently detect genetic differences between the cases and the controls. So the other talks that we heard, the in the genome, but give me the difference. Another compared to the encoding that we heard is that for DNA methylation, there's a huge variety of methods published and being used. In addition to how do we analyze the data, is what we use to perform DNA methylation mapping on a gene. For modification, this is quite straightforward because only 6 have a method. But here you see that a dozen methods exist that are distinguished how many samples you can realistically analyze and how CPG the genome these methods cover. Of course, we want to be covering the entire genome in as many samples. On the short term, what we did was select different technologies are as far to the top right, and which are candidates for performing associated studies. So in the next few an experiment where we actually tested all these different methods experimentally on a small number of samples to evaluate their performance, and at the same time develop a bioinformatic approach that can perform differential DNA methylation calling based on each of the different methods. So these were the methods we included. MEDIP and MethylCap are technologies that are very similar to ChIP-seq. They use an affinity purification step in order to enrich for certain parts of the genome that carry high levels of DNA methylation, and then sequence and align back to the genome. In contrast, RBS, reduced representation bisulfite sequencing, and the Infinium assay use bisulfite conversion as a way of converting DNA methylation information into SNPs in the genome. And while the Infinium assay uses an epigenotyping microarray, basically a SNP microarray that's adapted for DNA methylation, reduced representation bisulfite sequencing uses a way of essentially sequencing of CPG-rich genomic regions. And in order to provide a fair comparison of these different technologies, we applied them to 4 samples, each of the 4 methods, in particular a colon tumor and a matched normal colon tissue and 2 ES cell lines. And then we sequenced 2 lanes on the Illumina Genome Analyzer 2 for each of the methods. This also meant that we had to exclude whole genome bisulfite sequencing from this analysis because it's just fair to compare technology that requires 100 lanes per samples to a method that just requires 2 lanes per sample. And then we benchmarked each of these 4 technologies using a common bioinformatic infrastructure in terms of the accuracy of DNA methylation levels, the genomic coverage that these methods achieve, the ability to detect differential methylation with controls, and in terms of experimental efficiency. I'll focus here specifically only on the accuracy to detect differential methylation because these 2 are interesting. And visualize it, and you see a different character of the 2 different and methods, the 2 enrichment-based methods like ChIP-STAIN, whereas they use single CpG resolution CpGs. And when we compared the accuracy of the bisulfite-based methods, we saw a very high correlation in terms of the Infinium data versus the presentation bisulfite data. And what's very clear driver of accuracy for bisulfite is the genomic coverage. If we sequence deeper, we get very accurate DNA methylation, whereas we sequence only a very few reads or static noise. On the other hand, with methods, it became clear that there's a strong bias in the data. What coefficient run in the order of 0.6, 0.5 for MAP. That the false positives or methyl cap report as methylated, or they are mostly caused by high. This is a mention of the previous talks that repeat 10 previous peaks, and this is what we see here. We see a significant number of false negative genomic reads. Show any read enrichment, which are mostly due to pretty of these genomic regions. Peak in a specific region, either because there are no CpGs in this region, so that your antibody doesn't have anything pulled. Fortunately, both of these problems can be adjusted for using a linear model that corrects for this sequence-based bias. So we constructed a linear model that takes into account the number of reads detected by the enrichment-based methods, as well as input DNA sequencing and the CpG frequency, GC content, and repeat content of the region. And then we could correct for the sequencing coefficients that are most good as for best methods. And probably the most interesting topic is actually how do you detect differences between different samples. The unbiased approach, like pre-ENCODE, when we didn't know about the gene, is basically you look at peaks peaks that are different between the two samples that we see here. A couple of years ago, I experimented detection in order to find different peaks. But then, if you do peak detect this here and then on something like this here, you basically introduce basically discretize the data, and then may have a peak appear in one sample. So unfortunately, the basic assumption of our view of the world is not true anymore. It is not that we don't know quite a lot about the genome. We have a pretty good where genes are in the genome, ISCB islands, regulatory ones, and with all this, make pretty good assumptions in which puzzle actually be expect biologically relevant difference between the arcs. So this do statistics significantly, because if we switch our positions where we expect changes, we can just exact test to observe in one sample to the number of reads we observe, and not only treating each genomic region that was knowledge the genome as one unit. This is for we collect and publish literature sensors, regulatory regions, conserved regions. Then use this as the input for the different regions. What you can see here, what you see relatively good correlation between the measured tissue, but also some very clear. And because we sequenced data for 3 different technologies, we overlap between different technologies. In much further detail, but the overall conclusion was and RBS provide highly reliable data, whereas media acceptable to false positives and false negatives inefficiencies. And then the final phase, once you've performed such an epigenome association studies and you have identified the regions that are differentially methylated between cases and controls, the next big step is to prioritize these genomic regions for experimental follow-up. I don't really have the time to go into this in any more detail, but I want to quickly present 3 software packages that we have developed over the last 2 or 3 years in order to help biologists and especially people working in cancer research to sift through such candidates. The first one is this epigraph tool, which uses a large collection of genome and epigenome attributes, and in order to predict differences between types of genomic regions. So you basically upload a set of differentially methylated regions and the control set regions that are not differentially methylated, and the software will tell enrichment or deletion for functional elements, demethylation, conservation, analysis, but not focusing on genes, genomic regions, and aspects. And the second and third tool are who do DNA methylation develop class and moved on from the, it's provided a way of moving your analysis into a highly targeted process routine based. Quickly summing up my talk, we epigenome association studies provide a way of identifying disease-specific epigenetic alterations, and a technology comparison of DNA methylation mapping, and address the following bioinformatic to correct for sequencing bias that is present among methods, in particular repeat frequency. The detect differential methylation between control and statistical power. Here we use all the data that comes in order to help the software focus multiple testing correction, the statistical on a relatively small set of good candidates. And thirdly, the prioritization of candidates that could identify for validation. I think by switching from this genome-wide decision and unperfected computation and to on predefined regions, with that gives us and interpretation of the candidate. Hong San, Gu, and ING. Finally, I out the ESL number of reasons, but if you're interested in which is 23 or an update on that.
Talk that you exclude genome-wide pistol-fit sequencing isn't, but the three different methodology that you use is Epigenetic, while a lot of things using these technologies. So using pistol-fit sequencing, or
we have tried to say so we are doing,
especially in stem cell. I mean, you
very much genome-wide bisulfite sequencing, but for a different purpose. We use the whole genome bisulfite target reference maps of stem cells, for example. But this paper or this study was to establish genome-wide mapping as a way of analyzing large sample cohorts, and it's just not really at the moment to do 1,000 samples with whole genome biopsy. So just for the purpose of this technology, we excluded this method to keep the comparison to the methods. I would very much argue that it is better resources if you want to cover the new biomarkers, 1,000 cancer methylomes that are focal regions, than to do 10 cancer methods that are genome-wide. So it's basically just a cost.
Is it a Fisher's test to detect differentiate across your cases and controls, and that seems like a really natural approach. And I was just wondering, how would you incorporate multiple reps to that particular test? Let's say you were lucky enough to have 2 or 3 different samples from the same tumor. I don't know if that's even possible.
Yes. Yeah. So the situation we were facing, as soon as like number of cases and controls, we basically. But since you are asking about this, I could say if you look here, see that even in some heterogeneity, and basically what internal heterogeneity, another approach, every single CPG position, extension of fission with relation in order to give you an analysis rate for the. I think the point here is hypothesize that you know where things become so much easy, and that we are now in a position where things might happen in the genome.